Thank you, Lance. We're really excited to be here today. My name is Kathleen Mandziuk. I'm Vice President of Patient Strategy and Digital Health at PRA HealthSciences. Today, along with my colleagues, Nathan Smith and Kerry Deem, we're going to walk you through the tool that we created for the Fuse Innovation Challenge hosted by the FDA. We'll walk through the context about the challenge, the solution that we built and why we built it and the impact we feel it can have. And then Nathan's actually going to demonstrate how we were able to leverage the features of Neo4j in how we present our solution to allow it to be consumed by a larger volume and more diverse user group. So you may or may not know much about PRA HealthSciences. We're historically a contract research organization, or CRO. That's where our legacy began a little over 40 years ago now. And we really focused on oncology phase 2 and 3 research to help drugs become developed. Over time, we've grown quite large and also really diversified our feature set and our services that we provide in the healthcare industry. Through 2 major acquisitions, one being Symphony Health, allowed us to really enhance the secondary data assets that we have within PRA, as well as the ability to link our data sets using our Sonoma key with other types of data sets to really get a much more robust view of a healthcare patient journey. In addition, we've made investments in technology, such as our acquisition of Care Innovations with Bluetooth-enabled devices for medical use and research, also in a commercial environment. And we acquired a mobile health technology, Parallel 6, a few years ago. So all of those things have really allowed us to bring in additional expertise and additional tools in our toolbox to create solutions like the Ready tool that we will walk you through today. And our philosophy and our vision is really all surrounding the data and how it impacts patients and how we can leverage technology. That everything we do is about the patient, how we develop drugs, as well as how we get more insights for drugs that are already on the market. And that is really the focus of our that we will be presenting today. As most of you know, historically, clinical development, drug development has focused mostly on primary data collection. This is where we do things such as clinical trials and registries where we're collecting new data about a particular scientific question. However, our view of the patient healthcare experience is more robust and holistic, and the fact that we have access to secondary data, such as medical claims information, electronic medical records, existing registries, and other health economic information, really helps us enhance the visibility and the view and the understanding of the healthcare experience. And then we also complement that with data we collect, such as social listening and interviews and surveys. So all 3 of these layers of evidence give us a more robust and more knowledge about what's happening with treatments, either in development or commercial treatments. This is a team that we gathered across PRA. We come from a wide variety of backgrounds, and Kerry will talk a little bit more about the importance of having a multidisciplinary team and what that allowed us when we were developing this solution and building it specifically for the Fuse Challenge, but now we're also able to offer this to drug developers and other data scientists that could leverage the use of this tool. So the first thing we wanted to look at is to make sure what we've built is clinically relevant. There's a lot of things that we do with data that are statistically relevant or have an impact, but really is there a clinical impact to what we developed? So the real problem that exists out there with drugs on the market is polypharmacy. And the challenge by the FDA was to create a solution to help us have a more robust view of multiple treatments that are taken simultaneously from a patient? And are there potential safety concerns when patients are taking different combinations of products? So we spoke to one of our therapeutic experts within PRA, Dr. Fred Lewis, and he has a lot of passion around this. He's a psychiatrist, and many of his patients have been on multiple medications. And so there is a higher likelihood of a safety concern, either in a large group of patients taking the same medication, but in many cases, there could be a subpopulation that has the most impact or safety risk of taking combined medications. So it was great to have him as part of our team to make sure what we're developing does have real use out in the medical field. As you can see here from this article, FDA is really pushing to get more visibility around drug interactions. And the first time that a drug developer will see an interaction is in vitro when they're still developing the drugs. And as we start moving through the pipeline of a drug being developed, there are additional ways for us to get insights around potential drug-drug interactions. But as we know, clinical development is not the same as the real world. And that is really the challenge that we have as an industry, is measuring drug-drug interactions in a controlled environment, but then also getting visibility in the real world and what polypharmacy could really have an impact in certain patient populations. So as I mentioned, there's three main areas that we would typically identify harmful or risky drug-drug interactions. As I mentioned, being in the in vitro drug. So again, when they're developing it within the lab and they can combine different medications and see how they react, or within any kind of animal testing. And then when we move into the clinical trials, in many cases, we're going to exclude no known drug interactions from the trial or exclude the patients from participating if they are on specific medications. But in clinical trials, we have a lot of internal validity. So we know within a controlled environment with a very specific patient population, this drug is safe and effective. But as I mentioned, as products move into the real world, there could be other combinations. So all different types of patients are going to take different types of products that may have been excluded from the clinical trials. And really the only way we have today to get some insights around what happens in the real world in a post-marketing environment is through spontaneous reporting. So that is when a patient takes a combination of medication or even just a medication on its own and has an adverse reaction outside of what's in the labeling or expected. And they can report it, or the physician can report it to the FDA. So we end up with a very biased representation of those patients who actually had their information reported in the spontaneous reporting. We also have some post-marketing safety studies that we may do with things such as registries, but again, it's just those patients that participate. So we do have a gap in the evidence around drug-drug interactions to allow us to understand what impact it could have in a very broad, diverse patient population that was maybe not measured before. And that's essentially what this challenge was. I went to the industry for different organizations to leverage the tools and expertise they have within their company to come up with different solutions to be able to address this challenge. And so that's what we did, and we're really excited to walk you through exactly, what we put together and really demonstrate its utility for you today. Kerry?
Yeah, thanks, Kathleen, for giving that background. And you may have noticed in one of Kathleen's earlier slides, the diverse team that we had. When we were given this challenge, we were looking at, some of us were looking at, what can we do? What kind of solution can we come up with? And one of PRA's offerings is we have a couple different divisions, and we work, in our own, not silos, but our own areas, respectively, within the groups. So we started assembling teams from different areas, people with different backgrounds to come up with a really strong solution. And what you can see is we have, individuals from nursing backgrounds, epidemiologists, data scientists, machine learning specialists, individuals that have backgrounds in looking at data and digging into that data. So, we came up with this strong team to really explore and see what we could do. And we were excited to work together, not necessarily working together with individuals that we've work with in our day-to-day processing of what we do. So it was really, a diverse group to build and come up with a solution. So with that, I'll hand it over to Nathan, who really, dug into, the data and came up with a unique solution for us to show everybody. Nathan?
Thank you, Kerry. So our solution really stands on the shoulders of the hard work of some excellent data engineers at PRA who assemble a really amazing data set that allows us to have this commanding view of the U.S. healthcare market. We looked at data from February through April of 2020, and in that time frame, we saw 160 million patients, 1.1 billion prescriptions, and 640 million diagnosis codes. So out of that very large data set, we focused in on some smaller populations. Specifically, we looked at 293,000 patients nationwide who were taking chloroquines without a prior diagnosis for an autoimmune disease. And we also looked at a special subset of 1.7 million patients in the New York, New Jersey, and Connecticut area for our network analysis. And so that was the population that we loaded into Neo4j. We started from a data model, and it's a pretty straightforward picture here. You can see that these pink nodes represent a drug that could be prescribed, and then we track the date that the drug was prescribed and also the day's supply that was filled. And we can infer from the fill date and the day's supply that these two drugs might have been taken in combination. And then following the prescription dates, we look and we see a diagnosis date here that leads to a follow-on diagnosis. And so if we see this particular pattern happening a lot, that could be a potential sign that we have a drug-drug interaction here. And the nice thing about doing this in Neo4j and having this visual model is that it's really consumable by everybody on that diverse team that Kerry was talking about. Whether you're an MD or a machine learning engineer, we could all wrap our heads around this, and this gave us a common point for conversation and for beginning to work. And especially as we think about how important it is as we develop algorithms that they're explainable and it's not just a black box, having a very clear and easy to understand model like this helped us build a foundation for explainability.
Mm-hmm. And just one other thing I'd like to add to Nathan's comments, as you see here, due to the delay of the Fuse presentation, we had some time to switch gears and really focus on the COVID pandemic. And being an epidemiologist myself, I was very, very curious to really see what the data told us. So this is, again, just one example that this can be done with any combination of products. And the fact that the FDA had hydroxychloroquine approved for COVID over a period of time, that gave us this really interesting and unique opportunity to explore that facet in a little more depth. We're doing a lot of COVID research, and we felt that this information could be really widely used to help us understand the safety of that product, given that there's a lot of publicity and a lot of media attention with this product. And we wanted to take a more scientific and rigorous look at this current environment. So again, that's what Nathan will walk through today, this specific example, but we can do this with any combination of products. So.
Absolutely. Thank you, Kathleen. So yeah, in this picture, we're starting to show how we've taken some real world data and filled it into that model. And you can see that in this particular case, we were focused on things that might be related to COVID-19. So from that model, we then aggregate the data together. And that's important for a few reasons. One of them is for patient privacy and data security. And we really appreciate the advances that Neo4j has made with the 4.0 release in having those really granular security controls, because that's really essential in our business that's very highly regulated. We don't want to present data at such a fine level of detail that you could accidentally re-identify a patient based on their unique combination of prescriptions and diagnoses. And so here, these nodes represent everybody who's taken a particular drug, and then the purple node represents everybody who's taken a particular combination of two drugs, and the green node up here represents everybody who's had this diagnosis. And then we can put properties on these relationships that show how many people who took this drug then followed up and had this diagnosis. And so based on that information, we could identify a few cases where we saw that the number of people having a diagnosis following a combination of drugs was significantly higher than the number of people having that diagnosis following either one of the parts of that drug combination. And so here we have an example of when people took either hydroxychloroquine sulfate or chloroquine phosphate together with benzonatate, we saw a higher rate of tachycardia than with either one of those two prescriptions by itself. It's also nice in Neo4j to be able to represent hierarchies. And so the ICD-10 codes, which represent the diagnoses, come in a hierarchy. And when we looked at the category of all abnormalities of heartbeat, which includes tachycardia, but also brachycardia and a few other abnormalities, we saw that ceftonir, in combination with the chloroquines, had a higher rate of diagnosis within that category versus within an individual diagnosis. So several interesting patterns that start to emerge. And as Kathleen said, we can apply this same methodology to all kinds of different types of populations. So another real advantage that we saw by doing this work in Neo4j was that it allowed us to detect communities within our graph. So we could find people who had similar diagnoses and similar prescriptions, and then look at that drug-drug interaction within the context of a particular community. And particularly when we're talking about COVID, when we saw a community of people who have all similar diagnoses and similar prescriptions, and a high proportion of them have a COVID-19 diagnosis, the other people in that community potentially represent undiagnosed patients who might just not have the COVID-19 diagnosis, but have very similar clinical outcomes. So in order to develop those communities, what we're showing here in this picture is that we turned to the Graph Data Science library, and our first step was to run the node similarity algorithm and to mutate the graph based on the results. So that's what I was trying to show here with this picture with the dotted arrows, is that we didn't actually write the relationships that we just that we identified back into the graph based on the node similarity algorithm, but we modified the in-memory graph to represent those relationships. And that was beneficial to us because it really helped us with performance, not having to write all those relationships for millions of patients back into the graph. And then based on the similarity results, we ran the Levan community detection algorithm and wrote those properties back onto the graph. And so now we can see patients who are in one community versus a different community. And if you would like a very clear explanation of the same type of community detection process that we went through, I would recommend, if you missed it, to look back and find the session from the Data Science Connections event called What is GDS and Neo4j's GDS library, because Alicia Frame walked through an example in a retail setting that was almost exactly what we did here. If you just change out patients for customers and items for prescriptions and diagnoses, you could apply the same thing that's on her GitHub from that session. Is very similar to the type of work that, that we could do here. So now we'll take a quick look at how this looks in Bloom. All right, so we'll come over into our Bloom environment here, and I've set up a few different perspectives. This first one that we'll look at is one that shows the aggregate relationships. And again, remember that we want to be very careful with patient privacy. And so by viewing the data in an aggregated format, it helps reduce the risk that we could accidentally re-identify a patient based on their unique combination of prescriptions and drugs. So my search phrase here that I've set up lets me find the top interaction candidates for a particular drug. And you can see it's helping me autofill because I'm not that great at spelling hydroxychloroquine. There we go. Good, I'm thankful for the autofill. And then it's taking me into the ICD-10 manual, and I've broken up the different diagnosis based on the chapters so that I can find, a particular type of diagnosis that I might be particularly interested in. And chapter 18 is the chapter in the ICD-10 manual that deals with irregular lab values and results. And so here I can see I've got several different possible interactions that are coming up, and I'm just going to kind of drag these around to straighten out my picture a little bit, because I'm especially interested in this one that relates to tachycardia. And so now I can see my pattern here. I have the pink nodes represent all the people who took hydroxychloroquine or benzonatate, and if I double click on one of them, I can tell, wow, there's 338,000. Oh no, I'm sorry. My patient count, 405,000 patients who took benzonatate. And then I can also click on the relationship, and I can see a property there that tells me out of those there were 1,000 people who had that follow-on diagnosis of tachycardia. And I can explore, this purple node represents the people who took both hydroxychloroquine and benzonatate. And then this relationship represents that potential interaction that we might want to investigate further and follow up upon for hydroxychloroquine plus benzonatate leading to tachycardia. And I can explore lots of other possible combinations that might relate to palpitations, or I could explore different chapters in the ICD-10 manual through that search phrase. I didn't have to write any cipher, so that's really helpful to exposing this to people who don't know a lot about cipher, but they do know a lot about potential drug interactions. So I'm going to go ahead and clear the scene now and switch to a different perspective. And this perspective is where we can explore those communities that we discovered through the Graph Data Science library. I've set up a node that represents the biggest communities that I found, and the size of the nodes represents the number of people that are in that community. And so if I just pick my biggest community here and expand it, I've set up relationships that show the top drugs or diagnoses that were related to people who are part of that community. So if I expand this one, I can look and I'm seeing things like methotrexate and prednisone, omeprazole. This is suggesting to me that these might be people who are taking hydroxychloroquine for autoimmune problems. And so the population that's represented here might have a different type of reaction to polypharmacy because of their other factors like autoimmune issues compared to people in a different community like this one. If I expand this one, I can see these people also are taking hydroxychloroquine, but a big difference here is that they also have either a diagnosis for, this is the COVID-19 ICD-10 code there, U07.1, or this is one for contact where people maybe don't have a COVID diagnosis, but they know that they were exposed to somebody who did. So this population seems like they're definitely different based on their diagnosis. And then if I look a little closer, I see that they also have metformin and blood sugar. So this seems like people who might have a comorbidity of diabetes. And so we did some really interesting ideas of how we could explore these communities and to look at some different hypotheses for how drugs might interact based on additional factors that we discovered through the Graph Data Science library. So with that, let's take a look back at our slides and wrap up the presentation. So as we think about where we'd like to go next with this project, graphs are so good at including context, and there's other pieces of context that we would like to include in our analysis, like age, gender, ethnicity, other confounding factors, comorbidities, combinations that go beyond just 2 drugs, but possibly 3 or more drugs, and also broader searches across time and geography. And we'd also like to incorporate machine learning methods that infer drug-drug interactions from other known interactions. There's some really interesting work that has been done in that way that can complement the type of information that we're able to find from the real-world evidence. So we're excited to take this project into the future and partner with others to really make a difference for patients. So that brings us to the end of our slides, and I think we're ready for any questions.
All right, great. Thank you so much. That was awesome. So attendees, just a reminder, go ahead and type your questions into the Q&A portion of your big marker control panel. We're going to take a couple, and then anything that we don't get to, someone from either the Neo4j team, someone from the Neo4j team will reach out to you with the answer. We'll coordinate that with the PRA team. So let's go ahead and take some that have been coming in as you've been going. Could this tool be used to identify possible beneficial drug interactions in addition to adverse interactions?
I can take that one. That's a great question. And absolutely, that's actually something we were discussing at the Fuse challenge with the FDA, that there could be unknown beneficial. And we've seen that historically with different products that are combined, where a manufacturer may want to have explore additional research to determine if there are some benefits and better outcomes when there are some combinations. And we see many times that we have complementary labeling, where certain manufacturers will team up on products to co-market and co-develop and co-label the combination of those products. And again, the fact that we can see how products are interacting in a very wide, large population compared to the small groups that we see within clinical trials, it gives us the opportunity to really use some of the things like Nathan mentioned around AI machine learning to find patterns that we never knew existed before.
Awesome. Okay, and then a question about the follow-up steps. So after you've identified a possible drug-to-drug interaction with the ready match, what happens next?
Yeah, so we're continuing to develop this tool. Again, it's based on secondary data, as I outlined in the beginning of my layers of evidence. So again, it gives us a lot of external validity, but it doesn't actually measure within a controlled environment. So it all depends on the level of severity of the event. We would obviously work with the manufacturers to do any additional research that may be needed. And in many cases, there might be something like a registry or a post-marketing study that will follow patients taking both in the real world and collect additional details from those patients on top of the secondary data. So really complementing it with primary data research and that complementary data that I think will give us a more holistic view. And in those cases, there's a lot of different directions that a manufacturer could take at that point. Will it be added to their labeling, or, is it been shown that it's only within a certain group of patients? So there's a lot of different directions, but essentially, using this as a springboard to generate hypotheses that would typically then lead to additional primary research.
Okay, awesome. And then who are the end users? Who's the target audience for a tool like this?
Sure, I can take that. Okay. So mostly it's the drug manufacturers or the sponsors that are coming up with this product to be on the market, but it could be prescribers and people taking a look at these drugs so that they know, when they're giving out these drugs to the individuals coming in, that they could, realistically be hurting them or helping them. It goes either way. We could possibly help or hurt these individuals, but as Kathleen mentioned, we'd have to do further research. This is just to identify that these could be possible interactions, and then take it from further and further study and make sure that things are going on. But there's a couple of different audiences overall.
Yeah, and in addition to that, there are different industry groups or regulators, like the FDA expressed a lot of interest, epidemiology groups, the health outcome groups, payers, like insurance companies could be also interested in utilizing a tool like this.
Great. Okay, and then I think we have time for one more question. How long did it take to develop this tool?
That's a great question. So this really came about in the context of the Fuse Innovation Challenge, which was a limited time frame. And so we really had to work quickly, and we had excellent support and partnership from our friends at Neo4j. I could name a few specific names, but we really appreciated the collaboration that we had. So we put all this together in about 3 weeks to get to the very first stage of visualizing the data in the graph. A lot of that time was spent on just moving the data from our Hadoop environment into Neo4j. And then once we had it there, making sure that we could run the graph algorithms. But we were really pleased that we could take this from an idea, could we do this in a graph, to actually having a working prototype in just 3 weeks. And of course, no project is ever done. So we'll continue to enhance and continue to work on this for a long time to come.
That's awesome. Okay, great. I think that's all the time we have. So I'll turn it back over to Marilee. Thanks, PRA HealthSciences, for joining us today. This is a great presentation.
Yep,
Thank you.
Thank you.
Thank you all so much. It was a great way to start our connection sessions today. For those of you in the audience, if you submitted a question and it didn't get answered live on air, you can alternatively send an email to webinar@neo4j.com and we will get you pointed in the right direction. Stay tuned. Our next session is coming up featuring Demian Bolumio from Neoris and Patrick Wall from Neo4j, talking about using Neo4j 4.0 to maintain security with COVID-19 health check. So stay tuned for that. And again, thank you to everyone at PRA HealthSciences for an excellent presentation. We really appreciate your time.
Thank you.
Thank you.